Okay, so probably ignore the title, but PROTACs are in there, but I thought I'd tell you about all the things we've been sort of up to and how within the ICR, which the Institute for Cancer Research, sometimes we act like a biotech, we look at this as a drug discovery project, but we're also part of the University of London, so therefore we also have PhD students, so we can go for more of the elaborate things going on. And really we kind of, especially as a medicinal chemist or a chemical biologist, depending on what you look at it, you kind of use targets as inspiration to try and solve problems and the way you do things. And this particular target is something that's been sort of floating around the ICR for probably 10 years, and it led to a lot of innovations or things we try to do in order to take it on. We've sort of read the recent JMed Chem describing what a great medicinal chemist is. If you find you've got a good target, you never want to let it go. And this is where we got to with this. So this is probably the most simple description of how HSP72 works, but in the interest of time, I have to keep it short. So it's an ATPase, it's a molecular chaperone, so when proteins become misfolded and the cell struggles to get back to homeostasis, HSP72 can stabilize those misfolded proteins, stop them aggregating going forward. As you can imagine, the interest therefore in cancer, where proteins are often misfolded due to the oncogenic state, inhibiting this protein seemed like a very good and interesting approach to sort of tackling that. Again, must be the simplest description of it, in which you just simply have this ATPase, hydrolysis of ATP to ADP. This lid region collapses around this misfolded protein and stabilizes it from aggregation. So in a cartoon-like crystal structure, you can sort of see this is the substrate binding domain, this is this lid that sits around the top of where the protein is going to bind, and you have this nucleotide binding domain with this flexible link between the two. And ATP sits in this pocket at the base of that particular protein. So I joined the ICR, we did what everyone else to do, this part's not particularly innovative, is that we carried out a high-throughput screen. So our internal compound collection is about 250,000 molecules, so we screened that, and we got no hits at all. So okay, that seems like this is going to be a challenge. So we did a fragment screen, about 3,200 compounds in our fragment library, and we get one hit out the other side of that. So it's quinazoline, sort of 1.4 millimolar-ish. We had quite a nice little counter screen to this where we mutated out this serine to block the binding site where the quinazoline goes. And that way we can try to pick out by SBR where non-specific binding can be, and therefore it's a good way of a triage for compounds you wouldn't really want to develop any further. And that was it from that point going forward. Now, as a drug discovery project within the ICR, so we published that story back in Scientific Reports a few years ago. But as a drug discovery project, it somewhat came to an end then, as you can imagine, because we didn't have where else to go. But as I say, we are part of the University of London, and maybe we can try something a bit different, and we can do that by using the work our PhD students can do. So if our standalone approaches don't work, but you still believe in the target, what can you do? And there are two sort of approaches we took to this. One was, say, with PhD students, which is about covalent inhibitors, and the second one is around about phenotypic screening, which we'll go into there, which then leads on to use of PROTACs. Okay, so the short story on covalent inhibitors. Whilst our screening collection has not found any tight-binding small molecules to inhibit HSP72 reversibly, you see it's literature. There's this compound, which was developed by Venalis, which actually is a tight-binding ligand for that. Now, probably we didn't have any nucleoside mimics going around in our library, so not too surprising we didn't get any hits from that if this is the type of molecule that's going to bind to it. So from the KI, I put this as less than 51 nanomolar. That's a reflection of the bottom end of the assay, but certainly in our hands, it's as potent as we can sort of measure it at. But when you put this compound into cells, you find it's greater than 20 micromolar. So we sort of set up a hypothesis for a PhD project that the poor cellular activity that we're seeing with this compound is due to the competition with ATP. So the ATP value is quite difficult to get with too much accuracy on this one, but we think it's about 1 to 5 micromolar, high concentration of ATP in the cell. You see this large offset, sort of classic kinase challenge going forward. So part of that kinase challenge is this recent renaissance we've seen in covalent inhibitors, where T790M mutant EGFR has been successfully targeted with covalent inhibitors, where they've been targeting a cysteine to overcome the greater affinity that the T790M mutant has for ATP. So this was a good PhD project, or we thought it could be a good PhD project, and could we push that forward? So HSP70 is an ATPase, but it also has very, very high affinity for ADP, which makes functional assays quite difficult to do. So what we've been using for many years is an FP assay. If you're a chemist, you like FP assays because there's no antibodies involved. It's very much a very chemistry sort of focused process. Now, if you don't know FP assays, pretty simple. You have an FP probe, which is a ligand that has to bind to your protein, headed by a linker to a fluorophore. We fire plain polarized light at this because it's tumbling slowly because the protein's so big. You get high polarization out the other side of that. If we add an inhibitor, we can displace this probe from the molecule. The probe's now out in solution where it tumbles quite quickly, and you get low polarization back at the other end. So quite a simple concept from that. Now, if we wanted to make a covalent inhibitor, so we could use that to measure the affinity of our reversible ligands, this is what we would expect to see going forward. This would tell us we had a covalent inhibitor. So you get a typical S-curve with an FP assay as you displace the probe at higher concentrations going forward. But as we increase the time period, what we expect to see is a shift to the left as your compound appears to become more and more active against the protein as time goes forward because you're forming a covalent bond and taking the protein out of the game and removing it from its reaction. So I sat down and sort of designed this molecule based on the crystal structures of compounds bound to HSP70, which I'll come back to in a minute. And the student took this challenge on. They're not the easiest things to make, nucleosides, if you've ever worked on them. So he did a real good job on trying to putting these things together. And this is literally the first one that we made. So he came along, and as you see, you get an S-curve. So this is the, we convert it to the Ki. So we get a Ki of about 17 micromolar after what we're calling T equals 0. That's not T equals 0, it's as quickly as you can measure at that time point. So that's kind of good. It does bind. We get that first point. We're going forward. Let's leave it a bit longer. And as you can see, as we left this molecule, we start forming what we interpret as the covalent bond, and we start shifting our activity to the left. Now, these are only apparent KIs. You can't have a KI for something that's forming a covalent bond because it's changing all the time. It's a time-dependent value. But nonetheless, this was pretty good for a first attempt, we thought, going forward. So you do these classic sort of validation experiments when you're doing this kind of thing. So if we take out the double bonds, so we can't form the covalent bonds, you can see over the same time course that there is no shift in Ki over time. And if we block the reversible binding, so we knew lots and lots about the binding mode of these molecules, these two hydroxyls are very crucial for how the molecule binds. So we block that, and you get no covalent bond formation going forward. So this was quite satisfying. We can also do this with intact protein mass spec. And as you can see, over a time period, we mix together the covalent inhibitor with HSP72. We start off here, and as you go backwards from blue to red to green to purple, we're losing the starting protein, and then we're forming the covalent adduct. Do the same experiments with our two control compounds and show that you don't form a covalent bond with them. So that was quite satisfying, and that was all, worked out rather well. So we'd shown, somewhat to our surprise, that at the first attempt we can make a covalent inhibitor of HSP72, but we hadn't shown actually which residue was crucial for this. Now, this is the crystal structure that he was able to get hold of. So we weren't able to get a crystal structure of the covalent bond formed. And at the bottom of this pocket, you see cysteine-17. Now, this was originally the plan. We thought we'd target cysteine-17. It seemed somewhat exposed to that, and we could design a molecule with a linker to reach that nucleophilic residue. Now, what we did was mutate out cysteine-17, and you find that you still form a covalent bond. So it isn't cysteine-17. We then mutated every single cysteine within the protein, and it was not a cysteine that was involved. So in the crystal structure, you see our electrophile was pointing towards this lysine, lysine 56. Highly conserved lysine is involved in the salt bridge that we sort of published on and discussed quite a lot of detail before. So the indication is it actually lysine 56 that is the nucleophile in this molecule, not a cysteine going forward? I won't be telling you about it if it wasn't the case. So we can mutate out lysine 56, and you can see now you lose the covalent bond formation, and you lose the intact protein mass spec evidence that you're forming a covalent bond. So this was quite interesting to us. We weren't imagining we were going to target a lysine. There aren't very many examples of targeting lysine out there for covalent inhibitors, but it gave us a way into HSP72, and it's given us a way into a number of other protein targets that don't have cysteine available for covalent bond formation and that type of inhibition. Okay, so that was what we did when we were, in terms of a PhD project, when we were stuck by our HSP72 targeting. As more of our biotech side of things, This is how we took it on as a drug discovery project. So what we said was, well, if we can't directly target HSP72, what if we stop HSP72 being induced in the cell? Would that be an approach that would get us around this problem? So we did this by phenotypic screening. So it has some advantages. You can be somewhat agnostic about how the whole thing works, but you need to have some idea about what's going on. So in the, again, very simple biology of HSP72 induction, it's controlled by this transcription factor called HSF1, which is sort of sequestered up with HSP90 in a cell until you add some stress. The stress causes HSP90 via an unknown number or complexity of post-translational modifications to trimerize. You then translocate to the nucleus and cause the transcription of not just HSP72, but a number of different heat shock proteins. So our idea, following the, is if we could inhibit these unknown post-translational modifications and how they control the function of HSF1, we could block the induction of HSP72. And so we would do this by a cell-based phenotypic screen. With our definition of this HSF1 phenotype, where we look for compounds that would block the induction of HSP72 following a treatment with a HSP90 inhibitor. So this is quite important. If you stress the cell in different ways, the regulatory mechanisms of HSF1 are different to each other. You won't necessarily see the same phenotype, or the same compounds will therefore cause inhibition going forward. So again, we've screened our compound library, so more than about 250,000 compounds, and this is on a cell-based phenotypic screen. And this is one of the hits that come out from the other side of that. So it's an IC50, so we quantify the level of HSP72, which I can't go into the details of using a cell-based ELISA, but it's come out with an IC50 of 2.8 nanomolar. So straight off the bat, you've got a low nanomolar inhibitor in cells without any further optimization. So it's extremely potent. You do some biology, which again I won't go into, to think actually maybe this is a novel mechanism of action, maybe something we might be interested in. So those who have worked on phenotypic screens, you sort of have a few choices at that point. You might often have compounds that are nowhere near as active as that in cells. You might have to do cell-based SAR in order to improve the activity of them. In this particular case, obviously, we didn't need to improve the activity. We needed to understand what was going on and improve the physicochemical properties of the compound. So you could look on it and say, we're going to focus entirely on target ID to understand how my compound is working. Now, because we were thinking maybe naively at the time of multiple post-translational modifications were leading to the control of HSF1, we were expecting polypharmacology would be needed in order to inhibit multiple proteins to regulate HSF1. If you get polypharmacology, does that make your life much more difficult with target ID? You could focus entirely just on lead optimization and say, well, my drug works like a treat. It has the phenotype that I'm interested in. Why don't I just continue on where I'm going? Or you can carry out both of these two ideas concurrently. Now, the way you want to go about that will be up to sort of the different details of your particular organization. What we did was carry out both approaches simultaneously. So cutting down about 6 years worth of work into 3 arrows is we did use cell-based SAR in order to pick apart the SAR and what points are important. As we sort of heard around making capture compounds, we're looking for broad SAR to try and find where points are to solvent, basically. Again, very simplified version. What we find is the benzodioxane end is complicated, and I'll come back to what I mean by it's complicated later. The 2 central amides are just essential, and we couldn't really do too much about those, and the quinoline SAR is very, very broad. So we interpreted the very broad quinoline SAR as therefore being a route into solvent. So about 400 compounds later, we ended up with this compound called 236, which was used as a chemical probe to investigate how this effect on HSF1 was carried out in cancer cells and sort of phenotype it had. It also has sufficiently good PK in order to carry out tests in vivo, and also to start to design target ID methods. From 236, we had another 250-odd compounds, and we got to the clinical candidate, which I can't show you at this point. But with the clinical candidate, we were able to show that this is a single agent, so we didn't have to add a second drug like we were doing in the original screen. But as a single agent, we could get regressions across multiple ovarian tumor types. From the toxicology, our therapeutic index are greater than 6:1, and the PK was sufficient to give you favorable dose predictions to man. So that clinical candidate is currently being progressed into phase 1 trials, next year. Also say we made 450 compounds after the clinical candidate because we weren't certain we had the clinical candidate, which is sort of an interesting point. So target ID, as you just heard about the capture compounds, there are lots of those methods out there now, and we've tried quite a lot of them to investigate what's been going on with this one, and they have improved a lot, but they still sort of have their challenges. So they can certainly be very expensive, they can be very time-consuming, and there are no guarantees of success at the other end of it. A lot of them sort of deal with trying to limit the number of false positives, but they can increase the number of false negatives going forward with that by the stringency in which you do it. Most of them rely on the design of a probe, which we were quite happy with because we understood our SAR very well, but most of them also rely on mass spec proteomics, which we have some concerns over some of the things you might miss from that. So from the number of different methods which we did, what we found was this protein called Pirin, and which binds to our molecules with very high affinity. It's a bis-cupin, which has these two different barrels at this end and this end, and it's a crystal structure with the compound bound to it. It's just the link, the solubilizing group is flexible, so you don't see it in the crystal structure. It's a metal-dependent protein, but we don't bind directly to the metal, we bind to water-mediated waters that are bound to the metal through that point. So Pirin was described in the literature as a putative transcription factor regulator, which sort of sparked our interest, but there's very, very little on it out there. And this is one of the risks of phenotypic screening. You might turn up with a protein target that everyone knows everything about, in which case that's probably the end of your project. You might turn up with a target that knows nothing about, which then means your biologist has got a lot of work to do to try to catch up on the lack of knowledge. So this is one phenotype that's been described for Pirin. So in the melanoma cell line, WM266.4, where Pirin is really highly overexpressed and also amplified at the gene level. From shRNA knockdown, you can show in this cell line that it inhibits migration of cancer cells. So if you go to our paper, you can see this is a video, but I wasn't sure the video would work here, so I just showed it as pictures. And this is a scratch wound assay. So basically, you have a monolayer of cells. The wound forms from a little needle that rips down through the middle, and then you can watch your cancer cells over time walk back. Probably a more technical description than that, but walk back over to and fill up the gap as they were there before. We had 100 nanomolar of our probe, and you can see you can block that. The graph is simply a quantification that the machine does to assess how much gap's left over, essentially over time. So our compounds could phenocopy that part of what was described by in the literature about Pirin. But ourselves, we knocked down Pirin, we knocked out Pirin, we tried to knock in mutants of Pirin, and we could not phenocopy the action of the compound. So the compounds have highly potent anti-proliferative activity, and knocking out Pirin does not recapitulate that. So our conclusion from that was, well, Pirin is certainly a high affinity target with recombinant protein. There must be a second target to go along with that. Now, at that point, we were expecting polypharmacology. It wasn't a huge surprise. I didn't realize that polypharmacology would be quite as complicated as it's turned out to be. But what we wanted to be able to do was pick apart how Pirin was or our binding to Pirin was affecting things in cells, and how the second target, therefore, what part of the phenotype could be assigned to that. So this then takes us on to PROTACs, until I get there eventually. So we aren't in PROTAC drug discovery. Our ideas for using PROTACs were as chemical tools, and this was our first foray into doing them, or using them. So there's been a lot more literature since we started this process. Maybe some of these questions have been answered. You have to make your own choice of that, but this is where we were at the time and the amount of information that was out there. So we said we're going to make a PROTAC for Pirin, and we're going to use that to investigate the action of Pirin within cancer cells. We were going to assume that we needed to form a ternary complex between our E3 ligase and our target protein. There's been some great work shown of crystal structures showing that you are forming a ternary complex between your PROTAC and your two proteins in that sense now, but we didn't know this at the time. We're going to use CRBN as our E3 ligase that we're going to target with that, largely because we had chemistry in-house relating around fluorescence. We're going to stick to standard medchem principles, which I'll come to the details of what we mean by that. And we're not going to try to optimize the linker length. The idea of this was not as a drug, it was to use as a chemical tool. We thought if we get to the point where we have an optimized linker length, we're going to be here forever, and that was not the idea. And we're going to assume passive permeability for all these compounds, which hopefully you'll see why you might want to assume that, or try to understand why these molecules should or shouldn't get into cells. Okay, so this is the first one we designed. So at one end, you have a Pirin linker, so a bit like the CMC compounds we've been talking about. So you have a Pirin linker that needs to bind to your target. You have at the other end, you have a fluminimide derivative that needs to bind to CRBN. And you need to design a linker to put those two together. We were quite naive. We kind of looked at synthetic tractability as being the easiest point of that. So we've got these two amides at either end of that point. So we knew how to put these two parts together. So we designed that one, synthesized the compound, which is not actually trivial. It's about 12 steps to put that together and quite low yielding. But we can make enough of it to give to our biology colleagues. So we made it, tested it, and it doesn't work. It doesn't work at all. So it doesn't have a phenotype, doesn't deplete Pirin, doesn't do anything. So this is quite disappointing, and there was plenty of discussion about this. Just call that a day. That's enough of that. Because we didn't know whether a PROTAC could work against Pirin. And that's a part of something that's still up for discussion, is what proteins can be amenable to PROTACs and which ones aren't. And I think that's quite important going forward. Nonetheless, we thought we'd give it one more go. We persuaded ourselves to give it one more go. And one of the things we found out about our particular PROTAC was that fluminimide is known to have low aqueous stability because the fluminimide itself gets hydrolyzed. So it had a half-life under the cell conditions. As a compound at this room temperature, it's perfectly fine. But under the cell acid conditions at 37 degrees, it degrades quite rapidly with a half-life of about 3 hours. So we came to the conclusion, either it's going to work quick, or it's not going to work at all. So it needs to get in the cell quickly. So this was our plan, and say this is our standard medchem principles, and it's all going to work fine. So our SAR told us that we could do something about this methyl group, and the change to that methyl group masked that amide and improve the permeability going forward. So we're going to do that with a fluorine and form a dipole in order to block the way that water interacts with air. We don't really need this amide. We'll put it in there because it makes life easier for putting the molecule together. Let's see if we can take that one out. So this is the second generation one. We've got a fluorine here, so we cyclized the piperazine to give that particular end here. And it works a little bit. So you have to go up to 3 micromolar, which if you know about PROTACs, that's a lot of PROTAC. We need to leave it for 24 hours. That's a long time to still be leaving it for. But nonetheless, it works based on this principle of trying to make these improvements based on standard medchem principles. So emboldened by this, you can go along and do this a bit better. Well, actually, let's try and sterically mass this a bit better we can than just with a dipole with fluorine. Why have we still got a carbonyl here and an amide? They're a bit rigid. What's the point of that? There's no need for that amide either. Even though to put them together, it does help the synthesis for that. So this is our third generation PROTAC. Now with the chlorine down here, all the amides sort of sorted out. You have to put this together by Mitsunobu, which has been the rod for everyone else that's tried to make a PROTAC after that within the ICR, because it's a complete nightmare. It doesn't always work. We don't always know why. But nonetheless, there's our third generation one. And you can show with SPR that it binds Pirin really, really tightly, get a lovely little SPR trace. You can show that it binds CRBN really, really nicely at the other end using an FP assay. And now you get the lovely hook effect. You're down to 2 hours of exposure of the PROTAC and only 50 nanomolar concentration. Quite interestingly, you lose the hook effect when you leave it for extended periods of time. We could dial down the PROTAC and see we're still even getting degradation down to 2.5 nanomolar going forward. So we're all very pleased with ourselves on that one. Now, what you can do with these and say why you want them as tools going forward, and again, this has been just shown a lot now in the literature, that PROTACs are more selective than their parent compounds. And our interpretation of this is what we see here. So these two biomarkers are from the M4 to protein response, and they're biomarkers we see with the parent compound going forward. Now, we're able to deplete Pirin at very low concentrations where we don't induce the biomarkers. So our interpretation as a result is that this biomarker comes from the second target, not from Pirin, and our PROTAC doesn't degrade the second target, it only degrades Pirin. So now we have a tool whereby we can investigate how Pirin works. The second thing we can do is to try to use the PROTAC to generate in-cell SAR. So if you compete out with the parent small molecules, we see quite a lot, then you should be able to rescue the degradation effect. So here we're seeing a nice degradation at 5 nanomolar of Pirin. By adding the parent compound from our high-throughput screen, we can show that we must engage Pirin within living cells. So this isn't just an artifact of the recombinant protein or a strange thing with the PROTAC. So we get nice rescue from adding the parent on that one. As I said, the dark stain is quite complicated, and this is why we never went too much into it. You can lose one of the oxygen atoms, and you lose a bit of cell activity, but still nothing too much. Still bind Pirin really, really tightly, as tight as we can sort of see. You move that oxygen around one position, your cell activity is just gone completely, but you still bind Pirin rather well. But if you then try to do the competition assay, if you see our blue one here, which has no cell phenotype in terms of antiproliferative activity, but still bound Pirin, competes Pirin out within the cell as you'd expect. This compound here, with the oxygen only one position next to it, which still binds recombinant Pirin rather well, fails to compete Pirin out within a living cell. So this gives you intracellular SAR based just on target engagement, and not based on phenotype for complex targets. And that's the end. I just want to really, there's loads of people obviously that worked on this project over the last 10 years, but I really want to thank two people. Johnny's my PhD student who's just finished, who did all the covalent inhibitors work with HSP72. And if you ever want to take on a phenotypic project, make sure you've got a very good cell biologist, because you're going to run a lot of assays and doing a lot of interpreting of them. So Sui is an absolute legend, how she did all that work. So most of the cell work was from Sui and all the Western blots. And thank you for listening.